In a powerful show of support for nature, nearly every government in the world united behind the CBD's sweeping biodiversity plan for life on Earth. They committed to halting the extinction of threatened animal, plant, and fungi species. And in an historic first, they also committed to helping those species recover, so that the life we share our planet with can not only survive, but thrive. Making these goals a reality will take all of us working together. Reverse the Red is supporting governments, conservation experts, and communities around the world as they join forces to activate tried and tested tools and tie global efforts to local expertise. All in a coordinated effort to assess, plan, and act for wildlife. Together, we can save species from extinction. Together, we can secure our planet's future. Together, we can reverse the red.
Welcome, everyone. We are so excited to have you all here. I see many of you are already introducing yourselves in the chat. Please do keep doing that so we know who you are and where you're joining us from this morning, afternoon, or evening, depending on where you are. We are, again, very excited to have this special session on biobanking for species recovery. I am Megan Joyce. I'm the Outreach and Engagement Coordinator for Reverse the Red. We are a global action partnership, and we are co-chaired by the IUCN Species Survival Commission and the World Association of Zoos and Aquariums, alongside our other governing council partners, Botanic Gardens Conservation International, Rewild, the Toronto Zoo, and ZSL. And we're really pleased to have the WAZA team here today putting together this session with us. And we focus a lot on Target 4 and species recovery and Reverse the Red, and biobanking and genetics is a huge part of that. And so we're again very, very excited for this session. We just wanted to take a quick minute to thank our financial sponsors for Reverse the Red. We wouldn't be able to do any of the work that we do, putting together this vast network, working with all of you, and hosting these webinars without our partners. So thank you again to these partners. And without further ado, because we have a packed agenda and I am sure you are ready to hear from our wonderful panelists, I am going to turn it over to Lois, who's going to lead you through our conversation today. Two quick housekeeping notes. You have the ability to enable AI translated captions through the bottom of your Zoom application if you choose. And as we go through, if you have any questions, please add them to the Q&A function at the bottom of your Zoom, and we will save them for our discussion that we will hopefully have time for at the end. So any questions, please let us know. Please continue to add your introductions in the chat. And Lois, over to you.
Thank you, Megan. Hello, everyone, and a warm welcome to today's Reverse the Red webinar focusing on biobanking and cryopreservation as essential mechanism for modern wildlife recovery. My name is Lois Lelanchon from WAZA, the World Association of Zoos and Aquariums, and I will be your moderator for the next 2 hours. And today we are unpacking an urgent mechanism for biodiversity conservation. We all know that ecosystems face unprecedented pressures, and biobanking and cryopreservations have become essential insurance policies for life on Earth. So our focus today is directly in relation with Target 4 of the Global Biodiversity Framework, which calls for urgent management actions to halt species extinction, maintain genetic diversity, and safeguard wildlife populations. So over the next 2 hours, we will travel across 4 distinct phases, from global frameworks to regional networks, moving into also cutting-edge cellular technology, and finally expanding beyond the animal kingdom to the rest of the tree of life. So let's begin with our first phase on global frameworks, looking at the micro perspective of how and why. And for this, I'm pleased to welcome our first speaker, who's a true pioneer in the field. Professor Oliver Ryder is the specialist group leader for the San Diego Zoo Wildlife Alliance. His distinguished career spans cytogenetics, genomics, and genetic rescue efforts to reduce extinction risk. Today, we'll discuss the foundational role of the IUCN-SSC Animal Biobanking for Conservation Specialist Group in creating opportunities for long-term population resilience. Welcome, Oliver, and the floor is yours.
Thank you very much, Luis, and it's a pleasure and an honor to be here to speak to everybody about this topic. I'm really delighted that Revive and Restore and WASA are producing this, and my slides visible? So hello, everybody, again, and as I said, I'm delighted to have the opportunity to do this. I'm often asked what is biobanking? So just so everybody, we start from the very beginning, we might consider it to be the systematic collection, preservation, and management of biological samples for recovery of species. This is conservation biobanking. Biobanking for conservation is a tool to help reverse the genetic diversity losses that are key to species sustainability in the current environment. The biobanking supports the international framework for conservation of species and ecosystems. It's already been alluded to in the video, in the statements from Luis, and countries are now providing their reports to the Convention on Biological Diversity, and this activity encourages the ex situ conservation, and biobanking is a key tool here. Biobanking is not just a theoretical undertaking. It's already giving benefits for all of life through work that's already been undertaken. Because biobanking is a backbone for genomic studies, it's of global importance as we undertake this new era in biology to understand life through the evolution of genomes. And because of biobanking and the availability of samples, for example, it was possible to predict something that wasn't obvious. For example, that deer can be reservoirs for SARS-CoV-2 virus. But so that really applies to One Health and pandemic and global health issues. But biobanking of living cells can also contribute to genetic rescue, as I'm going to talk about more. But a notable example is the endemic North American mustelid, the black-footed ferret. A new founder was introduced to the captive population through conservation cloning. A sample from a female collected in 1986 sat in liquid nitrogen for 40 years, and then those cells have produced 3 clones of that individual. One of those clones reproduced normally with a male ferret from the current population and produced offspring that are healthy and have themselves reproduced. So living in the cryobank as frozen cells, there are cells that are now grandmothers. So cryopreservation of living cells and the also the effort to save materials that aren't living through blood and tissue, and of course saving gametes as well, offers new possibilities for conservation. And together they synergize actions that are going to benefit all of life if we only undertake more systematically, thoroughly, and expeditiously biobanking. And just to point out that biobanking has been a concern of WASA for some time. In 2018, the resolution was passed recommending that members participate in efforts to biobank. And of course, through the ex situ management plans, through the breeding programs of zoos, we have, in the last half century almost, been working on linking ex situ and in situ conservation, using ex situ populations to conserve species, to lower their extinction risk, to work with species that became extinct in the wild, and then to support in situ populations. So this is done through species survival plans or management plans. And although these have been in place for some time and considerable work goes on them, they face one constraining reality, which is that small populations lose genetic diversity. So we're familiar with these kinds of plots that show that even if we undertake the most efficient breeding programs for our living collections, we're going to lose genetic diversity. We know, though, that genetic rescue of wild populations is a recognized phenomenon. Genetic rescue is increase of fitness from introduction of new unrelated individuals into small declining populations. So increase in fitness means longer lifetime, higher reproduction, greater survival of offspring. And examples of this exist, for example, with the Florida panther. But genetic rescue may be possible using advanced cellular and genetic technologies, such as artificial insemination, which has already been shown, such as somatic cell nuclear transfer, which is cloning, which has already been shown, and through induced pluripotent stem cells, which is an area of active research. So if we can consider that we can use cryobanked living materials to contribute to this one plan approach, this conservation paradigm, where genetic diversity is stored in living material in cryobanks, this genes can flow in and out of the freezer, and over generations, what that means is we can preserve up to 100% of the genetic diversity of the samples represented by the samples that we have banked and its population that it supports. So this is an unprecedented opportunity in the history of life. That sounds big, it is. And we have the opportunity to contribute to that now. But there's a lot of missing technology, and we have to keep things secure. But because biodiversity is declining, there's not going to be a better opportunity to conserve biodiversity by biobanking it than there is now. So that provides a great opportunity, which is the reason for the establishment of the ABCSG, the Animal Biobanking for Conservation Specialist Group. And we're going to put the URL for the website for the ABCSG in the chat, or you can use the QR code there. We have a most recent newsletter that's just posted to the website so you can track the activities of this specialist group. The purpose of this group is to establish a decentralized, uh, uh, distributed global network to share information and expertise, to establish facilities to preserve all kinds of biobanking specimens, but we're especially interested in promoting the development of the conservation of viable cells and tissues for biodiversity characterization and conservation. We're going to hear more about this from Dr. Heinemann. But I would say that the core message that I have to deliver in this overview is that biobanking is an essential conservation tool. It affords options for the future that would otherwise not be available. And we have the opportunity now to greatly expand the efforts that have already begun in this area, in this realm, through the expansion of this decentralized, distributed global biobanking network, and facilitating the further work that's necessary. One of the things that is crucial to the development of this remarkable accomplishment or potential or utilitarian activity of all humankind will be to undertake this effort in a way that's inclusive and just, that benefits all of life, all of the world. And that is considered in the Kunming-Montreal Global Biodiversity Framework, which calls for not only undertaking these efforts, but it calls for doing it in a way that provides benefits in a just and equitable way. And one way of doing that is by incorporating notifications. Zoos have a great opportunity to contribute to ethical biobanking by sharing their information and biobanking resources with countries, local communities, and indigenous peoples from whose lands the founder animals from our conservation programs were taken. Wildlife conservation biobanking can provide new approaches for benefit sharing and adopting approaches that are responsible, respectful, and potentially collaborative. Thank you.
Thank you, Oliver. I cannot think of a better introduction to this webinar. And so thanks so much for framing the conversation and also highlighting how biobanking is such an important conservation tool. And precisely in your talk, now we'll look at how we can scale these capacities globally while honoring the local needs. So for this, I'd like to welcome Katie Heinemann, Conservation Program Leader for the IUCN SSC Center for Species Survival Biodiversity Banking at the San Diego Zoo Wildlife Alliance. Katie's background is in living cell conservation, and she works closely with the San Diego Frozen Zoo to expand these tools worldwide. Today, she will discuss mechanism for scaling genetic preservation globally, while simultaneously empowering and aligning with national conservation priorities. Welcome, Katie, and the floor is yours.
Thank you so much, Lois. I'm so sorry that I forgot to unmute myself, but one moment while I bring my slides back up. Yes, thank you so much. It's so wonderful to speak with this community today. And like Oliver, I am coming to this community from the perspective of the San Diego Zoo Wildlife Alliance and have the honor of representing the San Diego Zoo Frozen Zoo, which is really just one type of biobanking collection, but a very special one in terms of the living material that it represents. This collection was started in 1975 by Dr. Kirpanushka and has been a living repository that now contains over 1,300 subspecies of animals from over 11,000 individuals. This
Katie,
material.
apologies for interrupting. We can't see your slides.
Oh, I'm so sorry. Thank you.
You know, technology is always like that.
What about now?
We can see your speaker notes.
You can?
So we can see your presentation, but in speaker notes, with the speaker notes. Here we go. Perfect.
Okay, sounds good. We're going to go without notes. So you're going to get the unfiltered version of this presentation now. Yes, as I mentioned, the Frozen Zoo is an amazing collection and curated by an amazing team, including Marlis Hauk, who has worked for over 30 years in preservation of this collection. It includes a vast array of birds, mammals, amphibians, and reptiles, and new technology is continuing to evolve, which has allowed our teams to add new entries from the tree of life, including oaks, insects, marine invertebrates, and kelp in recent years. And through 50 years of biobanking, our organization has expanded to new types of samples and new areas of the tree of life. In addition to the Frozen Zoo, the San Diego Zoo's Wildlife Biodiversity Bank contains a native plant gene bank representing the native plants of San Diego County, a clinical repository of blood and serum, a disease pathology archive, a DNA and tissue bank, which represents an extremely wide variety of genomic information, and a wildlife artifacts collection as well. And through our work, we've seen how this biobanking information and collections can support global biodiversity targets. Perhaps the most obvious is Target 4 for halting extinction and protecting genetic diversity. A very impactful example of this is there's two species in the Frozen Zoo, or two individuals that represent extinct species, including the pauli, which is a Hawaiian bird that went extinct, but continues to live on in some manner through its living cells cryopreserved in the Frozen Zoo. Also, the genetic resources and the sequence information that has been derived from these samples has been used to really have very impactful insights for the conservation community. One example is my colleague at the San Diego Zoo, Aaron Wilder, did an amazing study evaluating extinction risk on the tree of life from genomic samples generated from this Frozen Zoo and other collections, and also is using this information to inform the genetic rescue of Pacific pocket mouse in San Diego. And so through seeing how these materials and this information can support global biodiversity targets has really inspired a partnership between the San Diego Zoo and the IUCN Species Survival Commission for what we call the Center for Species Survival Biodiversity Banking. And this partnership really leverages the amazing global reach and expertise of IUCN specialist groups. And working closely, especially with the Animal Biobanking Specialist Group, our goal is really to help support a distributed network of biobanks. And so when we talk about distributed biobanks, what is really the benefit of biobanks being distributed across specializations across localities? And so one reason for this is duplication. Biobanks are very valuable physical resources. And one kind of centralized biobank that's often invoked and is very important is the Svalbard Food Security Seed Bank in Norway. And this is an amazing resource, and the seeds are still very safe, but this is a physical location that faces threats such as glacial melting. And so having mechanisms for us as a global community to distribute these resources is incredibly important. Also, specialization and innovation can really come out of distribution of efforts and specializations. So here I'm showing in the middle column the difference between processing orthodox seeds and culturing culturing fungus for biobanking. These are very different processes, require different expertise, different facilities, and really it takes a global community to biobank the tree of life. And then finally, having a distributed biobank really centers the biological material where there's local knowledge and expertise. On the right, I'm showing a wonderful herbaceous plant from Oregon in the United States, where the people who collect the seeds actually lovingly need to put jewelry bags over the flowers so that they can catch the plants. So having local knowledge about plant phenology and other things can really help support biobanking and really expresses the need for some of this expertise to be local. And so quickly, with the little time I have, I'm going to talk about some of our distributed biobanking efforts in the United States, which our center supports and the San Diego Zoo is involved with. And my favorite one, because I'm also a plant lady, Marcello, I know he's going to talk later, but I'm also going to talk about seeds just a little bit, is the California Plant Rescue Network. And so this was an initiative started by the California Botanic Garden in 2014 and is really fiscally managed and operated under the auspices of the Center for Plant Conservation as well. But this is a group of 10 botanic gardens in the state of California that received an amazing public investment from the state, $3 million to biobank and seed bank all of the 11,000 plus globally rare plants in California in 2019. At that point, only about half of the species were biobanked in seed banks. And then here, just 7 years later, as a team, as a collaborative of distributed biobanks, this network has been able to have a transformative change, seed banking over 90%, 97% of the massive plant diversity in California. So this shows that this approach can work. I also want to highlight that there in the United States, there is efforts for biobanks to support the Fish and Wildlife Service ongoing in terms of biobanking vertebrates. And so this has been a partnership that through Revive and Restore and Fish and Wildlife Service Region 2 has really led in terms of distributing sample kits to recovery programs throughout the United States to help biobank T&E species or threatened and endangered species that have never before been cryopreserved. And so I really want to acknowledge the efforts of Revive and Restore in acquiring funding for this program, Marlis Hauk for really providing the capacity building and support for this, and also Brian Smallin, U.S. Fish and Wildlife Service Region 2 for coordinating the sample collection. Here I'm showing the mountain graham red squirrel. Oops, I spelled her name wrong. But this is one example of a species that has been collected through this initiative and had its genome published publicly. And then finally, because this is the WASA audience and regional associations are an important part of this community, I wanted to highlight an effort that's really being led by the Center for Zoonomics and Zoo New England within the AZA to really coordinate biobanking in this space. And so there's a new initiative called the AZA Biobanking Alliance, and it includes about 10 zoos throughout the United States. And I hope that you guys will read our forthcoming article in Zoo Biology, which I know a number of the members of this team were involved with. And then taking this out to a more global scale, we really see opportunities for the IUCN community to support distributed biobanking on a global scale. And we're really looking as a starting point at 4 biodiversity hotspots for which the San Diego Zoo has partnerships in country for long periods of time, including Kenya, Vietnam, Peru, and Hawaii. And so the first step in really aligning these biobanking capacity building efforts with national priorities is supporting on-the-ground partners to have national strategic planning efforts. And so there have been 2 national strategic planning efforts this year through this program. The first was held in Binh University in central Vietnam. And this was an effort that included both really led by our partners at the Institute for Ecology and Conservation of Nature in Vietnam. And we were able to support members of the Animal Biobanking Specialist Group, including Oz, who will speak later, and also Manabu Anuma, who leads the Asian Biobanking Network, to really integrate advice from the international community with on-the-ground and in-country expertise. And then similarly at the Wildlife Research Training Institute in Naivasha, Kenya, we gathered a group of stakeholders to give input on the Kenya National Biobanking Strategic Plan. And the purpose of these plans are really to start a conversation about the capacities are needed to map existing liquid nitrogen and other physical infrastructure to define what's needed and to really start conversations about capacity building between international partners so we can facilitate that knowledge exchange. And then at the same time that we're doing these strategic planning workshops, we're beginning to work on implementation through in-country training in vertebrate cell culture. So here on the left, we have members of the IUCN Specialist Group and Marisa Carodi from the San Diego Zoo talking about the development of a facility in the WRTI in Kenya. And then here we also have Katie Thompson from the San Diego Zoo Frozen Zoo leading field sampling demonstrations and also laboratory mentoring in collaboration with partners from Mandai Nature. And finally, I know I'm probably out of time, but I just also want to highlight that we're really excited at San Diego Zoo to host training opportunities for partners from different countries in different types of biobanking. This is our 2025 Frozen Zoo Cell Culture Workshop, from which we hosted 7 participants from 6 countries. And we are also proud to partner with the Huntington and the amazing Raquel Fogado, who leads training opportunities in plant cryopreservation in partnership with our IMLS grant and the Center for Species Survival, which allows international participants to participate. So if you're interested in collaborating, especially on training and capacity building opportunities, please feel free to reach out to me, and I will pass it along to the next speaker.
Thank you, Katie. And it's really important to align biobanking with national priorities. And I think you've highlighted that beautifully. And that brings us to now the second phase of that webinar, where we really look at the power of collaboration, data sharing, and sample management. So to start this second phase, we're heading to Canada. And I'd like to welcome Dr. Gabriela Mastromonaco, who's the Chief Science Officer at the Toronto Zoo, your Toronto Zoo. She's a reproductive biologist with 25 years of experience specializing in assisted reproductive technologies, and she joins us to present on building a coordinated national cryobanking framework to actively operationalize the One Health approach to conservation. So over to you, Gabriela. Welcome. And you're on.
Excellent. We're here. Thank you, everyone. First of all, thank you for this opportunity to speak with everyone and to share a little bit about what we're doing in Canada. We're perhaps fortunate that Canada did not have a systematic strategy for protecting its natural genetic resources. And so we're hoping to step up to the plate there. When people think of Canada, they may not think of it as the biodiversity hotspot, as you think of some of the regions in the world, particularly in the tropics. But we do have our biodiversity challenges, and what you see here in each one of those blocks are 10 edge, it represents 10 edge of extinction species. So we've got our biodiversity challenges across the country, primarily on the West Coast, where there's a lot of use of our natural resources. And they're facing some more critical threats than perhaps the rest of the nation, but equally distributed risk across the country. And with the change in the global environment, Canada is in a phase of accelerating its natural resource extraction. So in an attempt to become leaders in critical minerals and energy and all the things that we need to support our future way of living, we are becoming more and more concerned about our biodiversity and offering the right tools to protect some of those natural genetic resources. So Toronto Zoo has actually managed a biobank of zoo-housed animals, individuals, for almost 40 years. We've got over 100 species, as I've mentioned, primarily from zoo-housed individuals. But a few years ago, with the changing dynamics in Canada, we recognized the importance of supporting our nation in developing this systematic strategy. So we rebranded as Canada's Wildlife Cryobank, and really started on a journey that I'm going to share a little bit about today. Now, you'll see that I intentionally use the word cryobank to differentiate from the biobanks. We've got a national biodiversity biobank, and all the other biobanks that are really focused. It's a terminology that's used heavily in human biobanking. It's used a lot for data repositories, for genetic repositories, but we're really focused on living genetic repositories, and I'll talk about that now. So our goal for the cryobank, this National Wildlife Cryobank, is to preserve the genetic resources of Canada's ecological and cultural keystone species and help deliver on Canada's 2030 nature strategy. And that's an important concept when you're talking with the government, and I'll mention that in a second. We focus on living cells, sperm, oocytes or eggs, embryos, tissues, gonadal tissues or somatic tissues, and of course the cell lines that we can generate from those tissues. And our path forward, the path that we've chosen to take, is to collaborate with government agencies and Indigenous communities to expand cryobanking efforts nationwide, advancing national biodiversity goals while centering Indigenous stewardship and sovereignty. So our national cryobanking strategy is not a shotgun approach. It will be a very responsible, targeted approach that will be done in consultation. And I'll give you a couple of sentences there. Our ethos is focused around what we call the 3 I's of cryobanking. And this is very important in a nation like Canada, and we hope it'll be important in other nations across the world. So this is the fundamentals of our framework, is that it must have inclusion. In Canada, it would be with our Indigenous communities, but it can be local communities in other parts of the world, depending on the makeup of your nation. And of course, this is about responsibility to your community. It's ensuring that there's prior informed consent, access and benefit sharing, and working on collaborative models or frameworks for data and sample sovereignty. And of course, you need integrity. This is with our conservation communities, is to establish, support, and align ourselves with international frameworks, regulatory standards, and to support accessible data archiving and sharing. And a couple of years ago, Toronto Zoo, we recognized the importance of this, so we partnered with Species 360 to expand and develop a biobanking module that can either be open access and you can share your information, or in our case, some of the work with our Indigenous communities where the information has to be restricted, you're able to do that as well. So please reach out to Species 360 if you have any more information. It's an incredible module, and we hope people start using it for their cryobanks. And of course, to not stagnate, to continue to grow, we need innovation. And that's working with the biotechnology communities to support and expand species-based research, protocol development, and most importantly, responsible sample recruitment. So kind of avoiding that shotgun approach if it's not necessary, and being really targeted about your need and your interest. So the journey that we took, I call it the ABCs framework, or the ABCs of cryobanking. And we started with awareness, and this is working with our government agencies at the provincial and federal level. It's Environment and Climate Change Canada for us. And it was educating on the one plan approach, sharing the value of having a cryobank as the connector between the in situ and ex situ population management, sitting at the table with the heads of the species recovery plans, those who are writing the plans, participating in the plans, and having the conversations to introduce cryobanking as part of the action items for the species. Building trust. That is working with our Indigenous communities, having the conversations, bringing the voices to the table. So that would be Indigenous elders, leaders, community members. We've got Indigenous lawyers and academics. And of course, we've held the first workshop with representatives from across Canada. And again, here we're trying to understand what stewardship means rather than ownership. That's a very important concept in Canada, that nobody owns, but that our communities are able to steward the genetics, and what that's going to look like in a data and sample sovereignty model. And so I get asked the question quite a lot. So what are your top 10 species, or what are your top 50 species? And my answer is typically, I don't think that's for Toronto Zoo to determine. And so one of our approaches is co-development, bringing the government, NGOs, Indigenous communities, academics, species recovery folks, bring them zoos, bring them all to the table to have the conversation of what are going to be the top 10, the top 50 that we're going to really focus on in the next 5-10 years. So this, what we call a national wildlife cryobank model, this framework that I've been developing in Canada. I've got a couple of really close collaborators that, we've known each other for a couple of years, and we decided it was time for collaboration. So at the IUCN World Conservation Congress in October, we announced this really important collaboration. So Toronto got together with Chester and Dublin zoos to unite for cryobanking, and to unite under this brand, this National Wildlife Cryobank brand, to follow the ethos, the 3 I's of cryobanking. And we are now Canada's Wildlife Cryobank, United Kingdom's Wildlife Cryobank, and Ireland's Wildlife Cryobank. And of course, we got Doctors Sue Walker and Andrew Mooney with us online today. You're going to hear a little bit more about them. But we are sharing this model with anyone who's interested. Connect with us if you want to be part of this collaboration. We're sharing protocols, techniques, training, troubleshooting, working on standards together, and really trying to do responsible cryobanking for the greater good. Okay, so I'm going to share very quickly in the couple of minutes that I have left the story in Canada. We have 2 very important species. They're socioeconomically important, they're culturally important, and of course one of them is the wood bison. You could see the historical range up here in the kind of pale yellowy green, but of course they suffer from a number of things: the disease, tuberculosis, brucellosis, that they inherited from domestic cattle, the fragmented populations, there's genetic isolation, there's some hybridization with the plains bison, and of course sociopolitical issues, because our Indigenous communities are so reliant on them for food security and for cultural identity. So a group of us came together, known as Bison Integrated Genomics. I've been working with Dr. Greg Adams at the University of Saskatchewan for over 15 years, but about 3-4 years ago, we decided to really do a shotgun approach at this problem. So we've got those that are working on the TB tests, tuberculosis tests, those that are working on vaccine development, those are working to enhance our bison SNP so that we can look at introgression and genetic identity and lineage and all of that. And then, of course, the Genome Biobank. And this is where I come in. I'm the co-lead with Greg Adams, and we're looking at developing all the assisted reproductive technologies really for the one plan approach, so that we can connect in situ, ex situ through a cryobank, and using the conservation herds at the University of Saskatchewan and Toronto Zoo as the surrogate females for the calves. So you can see us here. In the first 10-15 years, we developed the tools, working both herds at the 2 locations. We collected sperm, embryos, put them into a cryobank, have all the calves to prove that the techniques work. And at Toronto Zoo, we've got a couple of really interesting firsts. The first calf born from sperm frozen for 35 years. That's one of the longest wildlife sperm frozen that's been used to make a living offspring. We generate calves. Obviously, we want donor surrogate females, recipient females. And so we sex sorted sperm, and we've got female calves that came from that. We've got 2nd and 3rd generation AI calves, so we can do repeated AI through the generations. And of course, not just fresh embryo transfer, but vitrified and IVF calves, living calves. So we've figured out all the technologies. We've got the tools ready to go, from ovarian stimulation, artificial insemination, sperm cryopreservation and sex sorting, in vitro fertilization, embryo transfer and cryopreservation, and of course the cell-based technologies. We are working on reprogramming both through somatic cell nuclear transfer and generating iPSCs. So after that phase, it's time to go to the wild. We've done a couple of experimental trips where, you dart them from helicopters. You've got about 6 to 10 minutes to collect the gametes. And of course, when we enter the diseased herds, we've developed the protocols to disinfect the sperm and the embryos. We've proven it in the lab. It's tuberculosis, brucellosis, so we have to use a level 3 facility in Saskatchewan, and of course, put the clean genetics into the cryobank. And as I speak today, I'm here at this webinar, but really I'm supposed to be in the Yukon. The mobile lab arrived yesterday, and the team is out there to collect over the next week from wild bison in the Yukon. So we're in the wild stage right now, and hoping to start closing the circle of the one plan approach. And I'm just going to touch on this. It's a project we just started with caribou, but with caribou, we're doing it the other way around, which is that we're working with our Indigenous communities first, hunted samples only, and making them part of it. We hire Indigenous youth in the community to be our research assistants, and working with the hunted samples in minus 35 degrees Celsius weather. We're trying to bring the samples into the cryobank. So keep looking there for hopefully some success. There are way too many people to thank, but these are some of our the funders, my collaborators, and the grants, of course, but so many more people. And thank you all for listening.
Thank you, Gabriela, and thank you for being with us today and presenting on Canada's Wildlife Cryobank and the important partnership, obviously, with Chester Zoo and Dublin Zoo as well. So now we'll move and cross the Atlantic to look at the regional platform across Europe. So our next speaker is Anna Mekarska, the EASA Biobank Coordinator for the European Association of Zoos and Aquaria. Anna manages biological samples across many institutions to facilitate scientific research. And today she will walk us through how a regional collaborative platform handles sample management, but also data integration and multi-species conservation science. Anna, welcome, and the floor is yours.
Thank you very much for inviting me to present European Association of Zoos and Aquaria Biobanking Initiative. The next speakers, Walker and Andrew Mooney, they will present our cryobanking initiative. So EASA Biobank has been created by EASA members to store and release for research blood tissue serum samples for better understanding of animal biology. We store the samples in the biobank in minus 80 freezers and in dry storage. We are happy and ready to receive samples from more than from all our members. We are having now 400 members in 48 countries in EASA, but we are also happy to receive samples from other institutions and also from some field projects. We are using same module as Gabriela has mentioned, ZIMS, to store records keeping, and we are promoting daily sampling routine from all species, not only endangered, not only EP species, but from all animals at least once during their lifespan, but of course opportunistically according to the legislation in Europe. We are having a huge potential in EASA. In our member institutions, there are more than 1,300 threatened species living now, and already more than 500 are under the EP program, so the managed programs. We are working in close collaboration with the EP coordinators. EASA Biobank is not an institution, is not a company, is initiative. And our samples are stored in 4 EASA Biobank hubs in RZSS Edinburgh, in EZW Berlin, in Zoo Antwerp, and in Zoo Copenhagen. All work of the EASA Biobank is supervised by EASA Biobank Working Group, which consists of our colleagues from zoos, scientists, geneticists, veterinarians. We are very happy and proud that since last year we are members of Global Genome Biodiversity Network, and very soon information about samples stored in EASA Biobank will be accessible through GGBN database. We have developed and we are still updating documents and standards in our biobank. This is very useful for zoos to use always the same standards, same protocols, and sending samples always to one biobank hub. And they are sure that all the legislation is covered by our procedures. We are having also security backup system, which is also helping us release samples for projects, and we store information about our samples in ZIMS. This is maybe we don't always recognize how big tool is this database, because each sample that is stored in our EASA Biobank is linked with the animal which came from. So we have really a huge metadata linked to our samples that's very much appreciated by the researchers which are using these samples in the future. Where we are now, after 7 years, we have more than 31,000 samples in the biobank from 1,292 species. Majority of institutions that belong to EASA are already submitting samples to the biobank. Till now, 68 research projects has been approved, and we are releasing samples to these projects. It's not a surprise that majority of samples are coming from mammal and bird species, but we are very much encouraging now zoos to sample and submit samples to the biobank from reptiles, fish, amphibians, and invertebrates. Also, we are storing in majority whole blood samples and tissue samples, followed by serum, but we are open to any other samples that can be stored in -80. So not living samples are going to EASA Biobank. To showcase how the samples are used to support research and to support species conservation, both in ex situ and in situ, I would just like to shortly go through the projects that we are releasing samples to. So majority of projects are linked with the EASA ex situ programs. Because many of data in the stud books is not complete. So we need, with the molecular genetics, we are able to complete the stud book data. But also many projects are requesting samples from ex situ animals to run research projects or reintroduction projects in the wild. Here we have examples of the Asian elephant, addax, amu leopard, European wildcat, including Scottish wildcat, blue-throat macaw, hyacinth macaw, and other species. It's sometimes very difficult where to place the project in this category or previous one, because many of EEP projects are having direct conservation impact, and they are insurance populations for wild populations. So any molecular genetics discovery in ex situ population can be used in situ and inverse from in situ to ex situ. We are also releasing samples to projects that are developing genomic markers to study in situ populations, including full genome sequencing. We have already released samples to projects run by customs, for example, to combat illegal wildlife trade. And we are very happy, and we support zoo veterinarians to use our samples to develop veterinary research projects that can help save our species as well. This A picture of a lion is with the purpose here, because last year we received 3 project applications requesting samples from lions for North African lion EP, for this vitamin D deficiency in big cats, and also for the cancer resistance in species project. And this is highlighting how the biobank is important for zoos. So zoos are sampling animals only once. They are sending samples to the biobank, and we are releasing these samples to the research projects. There is no need to sample several times animals or to organize 3 separate transports from zoos to researchers. Till now, because we are young, we have 6 publications based on the samples from EAZA Biobank and many more in line for publication. I would like to invite you to work with us, so zoos just to contribute and send samples to EAZA Biobank, and researchers to come to us and request samples for use in their research projects. You can contact me or Christina Wilson, the EAZA Biobank Working Group Chair. Thank you very much.
Thank you, Anna, for presenting EAZA Biobank. And we'll stay on the topic of EAZA and to hear a bit more about what you're all doing. And for this presentation, we have a joint session from the 2 key leaders of the EAZA Cryopreservation Interest Group. You've mentioned it, Anna, but we're welcoming Dr. Sue Walker, who's the head of science at Chester Zoo with 25 years of experience in wildlife reproduction. And joining her is Dr. Andrew Mooney, conservation and science manager at Dublin Zoo and coordinator for the IUCN Center for Species Survival Ireland. So together they will share their insights on advancing cryopreservation practices, building shared biobanking standards, and bridging institutional gaps across network. Welcome, Sue and Andrew.
Thank you very much appreciated. And thank you for the invite and organizing this session. It's great to see all the speakers and all the initiatives that are going around globally. So Andrew and I are here today representing the EAZA Cryopreservation Interest Group, and kind of highlighting how we've kind of moved cryopreservation into the thinking and into banking in terms of our support of the species conservation efforts within EAZA. So EAZA very much recognized, oh, I don't know if that's working, is it?
Yeah, I think your slide froze.
There we go. That's it. Andrew's in charge here.
Right there.
Thank you, Andrew.
No, look good?
Yeah, perfect. Thank you. So I think kind of backing what a lot of people have already kind of talked about already, EAZA itself really recognized the need for cryopreservation and the banking of living material within the EAZA community. So cryopreservation or cryobanking is almost an additional type of biobanking, which we've talked about. It's almost under that wider umbrella of biobanking, but it's focused on preserving intact or living cells, somatic and reproductive material for potential future revival and use. So as Anna's lovely presentation highlighted, we have the EAZA Biobank, and the EAZA Biobank really was kind of the infrastructure for it, and the thinking behind it was really built around thinking about blood, tissue, and serum, and kind of that -20 or -80 kind of freezing. So when we started to venture into how can we help and support the EAZA community with cryobanking, it was really clear that the infrastructure and the design that we had set up for the EAZA Biobank wasn't really appropriated or dedicated in terms of cryopreservation resources, expertise, or storage facilities to kind of do that -196 type degrees and using liquid nitrogen. And that's where the idea around the EAZA Cryopreservation Interest Group has come from. So EAZA also recognized the value of cryopreservation in terms of our population management and in terms of the managing genetic diversity within the populations that we care for. So the idea that cryopreservation safeguards irreplaceable genetic material, helping to maintain those healthy, self-sustaining ex situ populations over long periods of time. And under the One Plan approach, cryopreserved genetic resources can facilitate that collaboration between in situ and ex situ practitioners, providing genetic resources that support that integrated conservation planning and population recovery and future species resilience. And the idea around cryopreserving high quality biological samples while populations are still extant and genetically diverse is really critical for preserving options for future conservation practitioners. And it was felt really that EAZA members have a key role and also a kind of responsibility to play in this and provide samples because they have access to thousands of species and those samples. And so they could play a key role in cryopreservation and preserving living material. EAZA also recognized maybe some of the ethical challenges and considerations around preserving living material. So in October 2024, EAZA published their position statement on the use of biotechnology and cryopreserved wildlife material for conservation purposes. So in that, there was a series of highlighted points. And really it was around that EAZA supports cryopreservation as a tool for safeguarding biodiversity and ensuring species survival. EAZA supports the advancement and responsible use of biotechnology to meet conservation goals. They recognize the need for more species-specific research to develop tools and methods. They recognize the complex ethical and legal concerns around living material, and also look to protect their living samples and not using them within de-extinction efforts. So there were several groups within EAZA that really kind of highlighted this idea of kind of really pushing forward genetic preserving living materials. So the EAZA Reproductive Management Group and the EAZA Biobank really were looking at kind of building this into decision making and making it part of standard procedures within EAZA. So part of when we have that opportunity to take samples, we think about preserving those living tissues as well. But in order to do this, we had to inform EAZA members about existing cryopreservation initiatives and the priorities of EAZA ex situ programs to help people think about sample prioritization and what to bank. Also to support EAZA members to have access to the resources required for sample storage and provide expertise and guidance in cryopreservation and techniques and guidelines. So the SIG, or the Cryopreservation Interest Group, was established under the governance of the EAZA Biobank. And really, as Anna pointed out, we were focused on the kind of -20 and -80, but to enable that cryopreservation storage, what we were trying to do was build actually a network of cryopreservation network partners. And those partners are like-minded partners with aligned goals and kind of that shared ethos with EAZA. And currently, as of today, we have 44 individual members within the SIG that are represented both from within and external to the EAZA community. So I'm going to hand over to Andrew now, who's going to talk about kind of the operation of the SIG and how we work and support the EAZA membership.
So the idea of how the SIG operates is relatively simple. Ultimately, we're aiming for samples to be collected from animals within EAZA collections, and then those are sent with material transfer agreements to these cryopreservation partners as part of a network. And from there, those samples can then be used for approved research and development projects and used for assisted reproduction to support population management, all of which will feed into broader conservation of wildlife in support of EAZA goals and supported by the Biobank Working Group and Reproductive Management Working Group. The SIG is still relatively young, but we are incredibly proud of the work we've done so far. The goal that we want for the SIG is that every single EAZA member country will have a cryopreservation partner within them that we can direct EAZA members to send samples for storage. So currently we have signed MOUs with 5 different partners and 15 more in discussion, with 5 MOUs being prepared as we speak, bringing us, if all successful, to 15 of 47 countries represented. And I think in comparison to somewhere like Canada, this is very important for us in Europe because the logistics of moving living samples across borders is a lot more complex and challenging bureaucratically. So we're aiming for at least one partner in every single EAZA member country. Now, to support the expansion of the network, the SIG has developed clear criteria for the selection and review of these cryopreservation partners, really trying to make sure that those partners are aligned with the EASA values and are applying biotechnology ethically using open access methods, all in support of wildlife. In terms of governance and collaboration, then, we would like all of our partners to demonstrate effective governance, transparency, information sharing, and collaboration with individual EASA members and wider partners. And then maintain the infrastructure, expertise, health and safety, regulatory systems, all of which apply at an individual country level to make sure the samples are maintained long term. Now, with these partners, these cryopreservation network partners, we have developed an MOU with them. So the idea is that every single partner signs a MOU with the EASA Executive Office to become an EASA cryopreservation network partner, ensuring alignment with the EASA goals, ethos, and overall partner criteria we've just mentioned. And this does align with the EASA position statement on the use of cryopreserved materials and biotechnology that Sue mentioned. And these 5 partners we now have are not static. They are reviewed annually at the moment to ensure consistent standards and alignment over time, and to really make sure it's a living system and that communication is quite consistent throughout the process. And representing such a diverse partnership like EASA also means that we feel a bit of an obligation to provide the safest possible standards and I guess support to the EASA members looking to engage in cryopreservation. To support the individual EASA members contributing the samples, the SIG has provided a template material transfer agreement. And this material transfer agreement is between the individual EASA member and the cryopreservation network partner. It doesn't involve the EASA Executive Office, but they are considered legally binding agreements, so they are very important when submitting samples to these partners. And these MTAs really focus on ownership and protection of the samples and any potential derivatives from those samples, which the EASA members are donating. The MTA has been developed in line with the EASA position statement, as we mentioned, and ultimately what we're trying to do is provide the best possible guidance and support to EASA members who may not be experts in cryopreservation. The SIG, I said, is quite new, but we are very, very excited to have already 5 signed MOUs with cryopreservation network partners. But we are very keen to develop that network much further. And as we said, even if all of the MOUs we're working on at the moment are signed, that's still only 15 of the 47 countries we need. So if you do represent an organization or institution within one of those countries which wasn't highlighted, please do reach out to anyone part of the SIG, but myself and Sue would love to have a chat with you about what potential there might be to get EASA samples and to sign a cryopreservation network partner MOU with you. Thank you.
Thank you very much, Sue and Andrew. It's really interesting to hear about the EASA cryopreservation network. So thanks so much for being here and for presenting about that. Now we're going to enter our next phase, which is about expanding frontiers and emerging modern tech. And for this, we now turn our eyes to South America. And I'd like to welcome Adrian Sestelo, who's a conservative and reproductive biologist with 30 years of experience in wildlife species, currently leading the reproductive laboratory and biobank at Buenos Aires Eco Park. His work focuses on cryopreservation-based assisted reproductive technologies to support species conservation across Argentina and Latin America. And Adrian will guide us through the regional landscape, but also detailing unique biobanking initiatives and infrastructure challenge, and his effort coordinating conservation and population management networks across South America. So, bienvenido, Adrian. Please go ahead.
Muchísimas gracias, Luis. Es un placer. It's a pleasure, and it's an honor to be here with such great speakers, and trying to tell you the experience that we have here in Latin America, particularly in Argentina and the Buenos Aires Eco Park, and the news from Latin America for biobanking for conservation. First of all, I want to make a framework why we need to develop biobanking for living materials, because human people grow extremely in the last years, and it gives the problem that there's a lot of extinction. It's because the people grow in the world. Specifically in Latin America, we are so special because we have 40% of living species, and we have 6 of the 17 megadiverse countries in the region: Mexico, Brazil, Venezuela, Colombia, Ecuador, Peru. But we are few people living here, just 9% of the world population. But we are poor in the context of conservation. The habitat problem in Latin America is we lost 1/5 of agricultural lands and 1/3 of the forests have been lost here in the region. 60% of global deforestation occurs in Latin America. Yeah. In the last 44 years, we dropped off 90% of the natural resources. Then we need especially to develop some kind of support for biodiversity conservation. And as some of you mentioned in the previous talks, there are some limitations that we know from the conservation initiative from the WASA for zoos in the world. Then we need to bring some solutions for diversity conservation, and biobanking is one of the most important things that we can do for now and for the future, because it calls us to maximize the possibility to keep in biodiversity and bioavailability of genetic diversity in biobanks. As Gary and Oliver said, biobank consists in preserve living tissues: sperm, testicular tissue, ovarian, oocytes, embryo, somatic tissue, fibroblasts, mesenchymal stem cells, into liquid nitrogen for several thousand years. That maximizes space, large number of species and individuals, as all of them can mention. Each individual survives time indefinitely, and the transport of reproductive material for population management, it's easier than the transfer and live animal between regions. But in Latin America, we have so many boundaries with a lot of different legal laws. Then we need to improve biobanking and genetic reproductive assistance. In the case of Buenos Aires Eco Park, during the last 30 years, we developed a lot of techniques for reproductive assistance for felids, for example, for maximized embryo transfer, artificial insemination. We gave birth the first individual, wildlife individual, birth by this kind of techniques in Argentina several years ago. And but we are incapable to do those kind of things by ourselves. We work with agreements with the most important universities from Argentina, other countries, including Europe, to develop that kind of techniques. For example, ICSI to pellets, in vitro fertilization for cats, cloning for different species. We apply this kind of techniques to produce embryos, to produce the knowledge to preserve this kind of materials. We share information and samples with specialists of different kind of technologies, for example, genetics to develop some kind of analysis of biodiversity from wildlife individuals, for example, mink for jaguar. We developed specifically protocols to preserve deer's sperm and transferring that protocols to the threatened species of Argentina and Uruguay, for example, in the case of Pampas deer. We developed rescue from wild animals dead in the wild. The samples were sent to the laboratory, and we preserve a lot of samples from these such important individuals. We developed the cryopreservation from some special birds, for example, the Cronin solitary eagle, a critical endangered bird of prey here in Argentina and Brazil. We developed some protocol for sperm cryopreservation in South American primates. We perform rescue from dead animals to produce embryos with a material at the biobank. Give some information to sequencing genomes from wild species, or use the materials at the biobank. This is the most important thing, using the material to collaborate with the conservation. Not only reproduction, with the reproduction, we could share information. You could produce some special information to manage populations, like sex sorting for endangered birds, or to produce regeneration cell therapy through stem cells for using it in recovery for special individuals from endangered species. But all of this is our experience here in Buenos Aires Ecopar for our laboratory and biobank, where we have all kind of equipment to produce that kind of information. But it is not the most important thing that I want to talk today or to give you today. The most important thing is to talk about the collaboration, as all the speakers said previous to me. And as you can see, Latin America is huge. We have an enormous problem with wildlife. And for this reason, after a couple of collaborations with professionals, with some conservation congresses, producers in Colombia or in Brazil, we finally, after a congress in Cali last year, we decided to start with a new initiative, because as I told you, we hold enormous biodiversity here in Latin America, but the biobanking effort in the region remains isolated and less developed. Then we decided to start that we call the Latin American Biobanking Alliance for Conservation, with representatives from Panama, Colombia, Ecuador, Brazil, Chile, and Argentina. And the idea is to work with not only with frozen or biobanking, just living material, is to work together with genomics specialists and build a network in the region to share and produce information specifically for species selected that need the support from the biobanking. Then this is the idea. The LABAC was conceived to address this coordination gap, not to build capacity because we built during the last 30 years, but to connect and align the substantial capacity that already exists in the region. Well, it's really new. Our next steps on the network and the alliance we will get a formal governance structure to the and character for the LABAC, and strategic plan with a clear short, middle, and long-term goals. Identify priority species, as I told you before, as a initial programs or project to share in the region. Standards and protocols sharing and agreed for common directions in between different laboratories, a framework for financial and operational sustainability, define reference laboratories and areas of expertise within the alliance, for example, for genomics, for cells, for sperm, for living material, for embryos. Build a training capacity building strategy, and the main problem just is to agree the approach for legal regulatory coordination in the region. But when we are going to talk or decide those next steps is in a couple of months after the first International Symposium on Reproductive Biotechnologies Applied to Conservation that will occur in August 2021 in Campo Grande, Brazil, we will have the first annual meeting of the Latin American Biobank Alliances. And fortunately, we have the support of Toronto Zoo, San Diego Zoo, the ABC specialist group of the IUCN on guide to guide our next steps. And this is the most important thing that I want to share. In the region, we are growing. We are starting with our alliance to perform the most important development of biobanking for wildlife species and trying to build that network not only in the region, just connected with IUCN, with WASA, with Reverse the Red. And for more information, just connect with me. Thank you.
Muchas gracias, Adrian, and thank you for sharing your perspectives from South America, the challenges, but also the opportunities, and letting us know as well as about the Latin American Alliance. So now our next speaker bridges the world of advanced medical breakthrough and wildlife survival. We're welcoming Dr. Ospomp, who's the lab head and also assistant vice president at Mandai Wildlife Group. As a cell biologist, especially in stem cells and embryology, he's leveraging cutting-edge cellular tools to advance endangered species recovery. And today he will discuss the evolving role of cellular biobanking, stem cell technologies, and advanced reproductive methodologies in turning stored samples into active recovery tools. Welcome, Oz. The floor is yours.
Okay, thank you so much for the invite. So Mandai Nature is the conservation arm of Mandai Wildlife Group, which includes Singapore Zoo and 4 other parks. Our common overarching vision is creating a world where people and wildlife thrive together. Mandai Nature aims to prevent the extinction of the most threatened species in Asia, with a focus on Southeast Asia. Our activities are very diverse, but today I would like to zoom in on one of our most dynamic field, which is the live cell biobanking, which I'm leading. So today I will introduce our biobank. I will talk about our biomaterials and then about our technologies and conservation applications. So our biobank currently contains more than 4,000 vials from 175 threatened species, including mammals and non-mammals, such as reptiles, birds, and amphibians. And we tailor the optimal conditions for cells from each and every species. Our biomaterials are quite diverse, including germline, tissue, somatic cells, as well as embryonic cells, intact embryos, and adult stem cells. We collect the cells from a variety of tissues, both post-mortem and non-invasively from live animal. Each cell type has pros and cons. So what can we do with these cells for conservation applications? So as was mentioned earlier, we can expand them, extract DNA, and there are many applications that we can use the DNA and the RNA. With the live cells, one of the applications that was also mentioned is reprogram them into induced pluripotent stem cells, IPS. We can introduce certain genes which make them lose their identity and become a pluripotent, which means that they can become any cell type in the body. We can collect skin cells, reprogram them into IPS, and then differentiate them into heart cells or disease-relevant cells. Another important property is that these IPS are immortal. They can divide infinitely, in contrast to the somatic cells. Very importantly, in our lab, we use integration-free method, which means we do not mess with the genome, which is safer for downstream application, but is more challenging because the reprogramming is less efficient. So we don't compromise on the quality. So far, we reprogrammed cells from 14 different species, and this is led by Dr. Chuye from our lab. The first species that were reprogrammed were non-human primate species, endangered and non-endangered. So what can we do with these IPS for conservation purposes? One application is disease modeling. So as you probably know, one of the most devastating diseases in the wild right now is the African swine fever, the ASF. When porcines that origin from Asia, such as babirusa, wild boar, and bearded pig, contract the ASF virus, it's a death sentence. They will die within around a week. By contrast, porcines that origin from Africa, such as the red river hog, are believed to be asymptomatic carriers, probably because they co-evolved with the virus that also origin from Africa. So we collected the cells, reprogrammed them integration-free into IPS, and then differentiated these pluripotent stem cells into disease-relevant cells, which are monocytes and macrophages. So now we have disease-relevant cells from porcines that are resilient to diseases and ones that are susceptible, which may serve as a platform to understand the basis for resilience for ASF and to model the disease in vitro, something that right now researchers do not have. And you can read more about it in our preprint. Another application, and it's an exciting field that is called the blastoid or synthetic embryos. So we used orangutan cells, reprogrammed them into IPS, and Nicole from our lab aggregated them in a special way that make them appear like a blastocyst. So on the left, you can see human blastocysts. On the right, the orangutan blastoids. These aggregates don't only look like a blastocyst, but also the cells within the blastoid adapt the identity of the embryonic cells at the right location, and they also behave like a blastocyst. For example, they show implantation properties. And again, you can read more in our preprint. So these are IPS, and these are just some of the applications. I think I would like to use this opportunity to also thank Revive and Restore that funds some of our IPS-related projects. So what else can we do with live self besides reprogramming? As mentioned earlier, we can also use reproductive cloning. So our embryologists, Kanchana and Hygiene, that left the lab recently, use interspecies somatic cell nuclear transfer. So the gist of it, we collect somatic cells from threatened species, and we need oocytes from a related species that is domestic, that the oocytes are available. For example, fishing cat and domestic cat as a surrogate. We enucleate the oocytes, and we introduce the nucleus of the threatened species into the empty oocytes. And at the right condition, it can develop into a preimplantation embryo that can be transferred to a surrogate mother or can be biobanked until further use. We can also use the same strategy for threatened porcines using domestic pig. So that's exactly what we did. Last year, we generated interspecies somatic cell nuclear transfer fishing cat embryos. And earlier this year, we repeated this exercise with bearded pig and a domestic pig. We also tried to do it with babirusa and domestic pig with less success, probably because they are too far away evolutionary. The final example that I want to share is gametogenesis. Gametogenesis come in several flavors. In our lab, we are starting to use primordial germ cells or PGCs. This is super important for avian conservation because in contrast to the mammalian toolbox where we can use the IVF, somatic cell nuclear transfer, and stem cell derived gametes, avian cannot use these tools. However, PGCs show a lot of promise for avian conservation. Therefore, Nicole and Kanchana from our lab went to our collaborators in Bangkok for Dr. Kanika, where Dr. Kanika taught them how to collect these very precious cells. So the gist of it is that the PGCs are born at the wrong location and they migrate towards the gonads via the bloodstream. So if we collect the blood at the right time window, we can collect the PGCs, which can be stored in liquid nitrogen for further use. And the beauty is that we don't need to sacrifice the embryo. If done right, the shell of the threatened species can be sealed and the embryo can be developed. So what can we do with these PGCs? We can transplant them to a developed chick embryo surrogate. And if the chick embryo will develop into a rooster, it will generate sperm of the donors. And if it's a hen, it has the potential to generate eggs of the donors. Importantly, in Madai, we have a very elaborate collection of threatened avians. This is just a snapshot of some of the interesting birds that we have in our zoo. So the idea is to collect PGCs from these threatened species for future generation of gametogenesis using chicken as a surrogate. If you want to learn more about our activities in Mandai Nature, please look for the Straight Time publication from last year, or you're more than welcome to contact me. Thank you very much.
Thank you, Oz. That was a really interesting presentation. And also that really unlocks the potential of stem cell biology for disease resilience. So I think that was extremely interesting. And as you have now seen, we've focused this past, I'd say, hour and a half heavily on fauna, but we're not forgetting also our friends from flora, because everything is obviously interlinked. And so this is why it's really important for us, especially as we're looking at the global biodiversity framework, to also have the perspective from the botanic gardens as well. So to close out our speaker presentations, I'm delighted to welcome Marcello De Vitis from Botanic Gardens Conservation International. Marcello is a plant ecology and conservation specialist dedicated to improving regional native seed supply chains. And he will expand our horizon today by discussing the vital role of seed banking and botanic gardens in global habitat restoration and also plant genetic preservation. So, Marcello, the floor is yours.
Thank you. Thank you so much, Lois. Okay, let me see. Share. Okay, please let me know if you can see full screen my presentation.
Yes.
Yeah, okay, thank you so much. Hi, everyone. And thank you, Lois and Megan and all the Reverse the Red team for the opportunity to represent BGCI here today and contribute to this webinar and represent the plant kingdom as well. We have already heard about plant conservation from Katie, so thanks, Katie, for introducing the topic and for sharing such interesting efforts in this area. My presentation is not going to be about a specific species, but I'm going to provide an overview of biobanking for plants, and particularly the role of botanic gardens in this specific topic. So, a little bit about Botanic Gardens Conservation International. We are a global member organization with almost 1,000 member institutions. And we were established in 1987. So next year is going to be our fourth year anniversary. And our secretariat office is based in London, United Kingdom, but we have also regional offices worldwide, like China, Africa, the US, and we also coordinate regional networks of botanic gardens in many regions of the world. Our mission is to mobilize botanic gardens and engage partners in securing plant diversity for the well-being of people and the planet. And so this is from our database Garden Search, where we map plant conservation organizations, and it is possible to see that around the world there are about 3,700 institutions dedicated to this particular area, holding collections of around 30% of the known plants currently inhabiting this planet. Ex situ conservation for plants takes the form of seed banking, tissue culture, cryopreservation, which represent what we usually think about biobanking is for plants, but even living collections could be considered as such if we think of them as field gene banks. And we're going to see a little bit about that later in the presentation. As most plants growing on this planet in this current geological era produce seeds, we tend to consider the seed as the most common unit or propagule to start with for plant conservation. Conventional seed banking and seed long-term preservation involve freezing seeds at low temperatures, as this slows down the metabolism of the seeds and increase their longevity. To safely freeze the seeds, though, we need to decrease their moisture content, otherwise the water would become ice crystals and would damage the cells. And to do so, we need to go through the critical step of seed dehydration. But not all the seeds can tolerate, unfortunately, dehydration because of their biology. Based on the tolerance of the seeds towards dehydration, we define seed behavior and identify orthodox, intermediate, and recalcitrant seeds. Orthodox seeds are those that can tolerate desiccation and that can be stored under conventional seed banking, while intermediate and recalcitrant do not tolerate freezing and desiccation, respectively, and so cannot be stored under conventional seed banking. In this slide, I share more details and info about conventional seed banking. And you can see here that I list the standard conditions that we use for dehydrating seeds and store them for short, medium, or long terms. Seed banking, which we also sometimes call seed storage, is a very critical step in a long process to collect, hold the seeds and then reintroduce species in the wild. And specifically, seed storage or seed banking come after a few more steps like seed collection, seed cleaning, seed testing, etc. And in each of these steps, we follow very rigorous protocols, often science-based protocols, to make sure that, for example, we are capturing a good genetic representation of the source populations, and we are maintaining it throughout the whole process. So if seed banking is not done properly, there is a very high risk to waste all the efforts that we have done until this point. So it is very important, since we have standard protocols and a lot of research into seed banking, it's really important to follow these standards and protocols. Worldwide, botanic gardens have been very active in seed banking. O'Donnell and Sharrock in 2017 were able to count 350 seed banking botanic gardens in 74 countries, holding collections of more than 56,000 taxa, including many rare and threatened ones. Botanic garden seed banks have been more traditionally playing the role of conservation seed banks, where we usually store small amounts of seeds of mostly rare and threatened species with the goal of long-term preservation, reintroductions, research, and also exchange with other institutions. And here we have on the bottom right of the slide an example from the seed bank of the Botanic Garden of the University of Ljubljana in Slovenia. And you can see within the freezers these relatively small samples, like all these little vials. They look small, but they might contain, depending on the seed size, they might contain thousands of seeds, each vial. But definitely we can see that, the samples are relatively small. While in more recent years, the concept of restoration seed banks was introduced, and which is used on the opposite to define seed banks that are more dedicated to storing large amounts of seeds for shorter periods of time and mostly apply to common species. And for this reason, on the bottom left, I am bringing this example of the BLM National Seed Warehouse System in Idaho. Which is probably completely at the opposite extreme of a traditional conservation seed bank, in the way that in this specific case, this is a governmental infrastructure, very big, and many botanic gardens and conservation NGOs might not have the space or capacity for these. But this is just to show that the concept of the seed storage for land, that it's more, for the purpose of land restoration. Smaller examples exist at botanic gardens as they are getting more and more involved in the important global goals for ecosystem restoration, and there is an increasing need for storing larger amounts of seeds of also common species, so not just rare and threatened, and sometimes for restoration purposes, this is for like shorter periods. On the other hand, tissue culture and plant cryopreservation are alternative storage methods for exceptional species that produce few seeds or seeds that are intolerant to drying or freezing, as we have seen in the previous slides. And these techniques allow to grow plant tissues on in vitro medium and then possibly store them in liquid nitrogen at temperatures below minus 130 Celsius degrees. Most protocols until now have been developed for commercially important species, and much research is still going on to adapt those protocols to wild rare plants as they have different and unique requirements. From BGCI's garden search, we know that 177 gardens indicate that they have tissue culture and or micropropagation facilities, which again highlights the important role that such organizations have in implementing plant conservation. However, these techniques require highly specialized expertise, highly specialized infrastructures, and greater resources than conventional seed storage. including financial resources, as these technologies can be quite expensive. So for those botanic gardens that cannot establish such laboratories and facilities, field gene banks or living collections can be the way to preserve the genetic diversity of those species that cannot be stored under conventional seed banking or do not produce seeds for some genetical or environmental reasons. And so this is about having probably, everyone who have been in a botanic garden have seen the living collections. Some of them are, just for display, but some other collections, maybe behind the scenes, living collections are there for genetic preservation. If we talk about large plants like trees, there are definitely some logistical limits, like the space limits. Not all the botanic gardens have the space to host large living populations of tree species, for example. So it is really important here that there is a good collaboration among botanic gardens and arboreta to maintain meta collections and exchange genetic material. And this is where a network of botanic gardens and plant conservation organizations, again, can play a very critical role. So with that, I conclude, and thank you so much, everyone, for your attention and interest.
Thank you very much, Marcello, for reminding us of the animal kingdom and the importance also of seed banking on that. So very much appreciated. And now that concludes our presentations for today, but we'll move now to a live panel with a few questions. So I will ask all of our speakers to please put their camera on if they can. And I will start. We've had a few questions in the Q&A box, but I would like to start with one question that I feel is very important. And as we're talking about obviously sample movement, I'd like to know what, from the policy standpoint, also from a funding standpoint, what are the most critical gaps that we see right now when we're talking about the transfer of samples and the shipment of samples between institutions and the biobanks, for instance. So I guess we can start. I don't know who would like to start, but any volunteer on that one?
I guess I can touch base on the IAZA Cryopreservation Interest Group perspective. When we were developing the MOUs with individual zoos, aquariums, and universities, the idea of cost is always very important. So we offer guidance on that on an individual case-by-case basis, where the material transfer agreements outline all of the costs and who's responsible for those costs, whether it's the collection of the sample, transport of the sample, processing the sample, or then the long-term storage and use of the sample. So that really does vary depending on who's submitting the sample and what they're willing to contribute and what our cryopreservation partners are able to provide in tandem. So there's no one size fits all for that. We kind of let everyone decide amongst themselves how that will work best.
Thanks, Andrew. I see Oliver's hand.
Thank you. Yes, Andrew is, of course, quite correct with I think the larger issue is between nation states, where the regulations of CITES and the Nagoya Protocol under the Convention on Biological Diversity come into play. These agreements are, compliant with global policy, but they turn out to be serious challenges for transfer of biomaterials between countries. And that's one of the reasons that having a distributed network is so important. While the nations of the world, through their treaties and conventions, explore how to lessen the impact of this issue, we're losing biodiversity. So, but within countries, the collection of samples and its storage can increase the global the global extent of the functional biobanking of biodiversity, while the methods of sharing it through the Nagoya Protocol and the other policy regulations are being discussed.
Excellent. Thank you, Oliver. We have also another question, which is when we're seeing, all of the progress and the advancement and the technology progress, as highlighted by the presentation of Dr. Pomp, I have a question for. We are really seeing that we're moving from just sample storage to STELSEM applications, and I would like to also know. What about those institutions that are smaller, and how could actually they catch up with this technology and all of that? So I'd like to open that conversation if anybody would like to start with this. How can we ensure that we're being inclusive with the progress of technology and the different resources that it might require?
I'll jump in if nobody has something right away. I think one of the important things for all cryobanks, but particularly the newer ones or smaller ones, is to really build partnerships. So there's academic approaches, there's whether you do it through a grant or a partnership with an academics, and other NGOs and other such partners, but really partnership, and look to your local university. They've got the equipment, they've got the technology, and they've got the trained personnel.
Thank you, Gabriela. Anna?
Yes, and this is exactly what we want to develop in EASA, because we have big zoos with high technologies developed with the big laboratories, but also much more smaller zoos. And this is how we plan to run the projects within the cryopreservation network. So smaller institutions, they can receive the protocol, minimum processing samples on their side and just transferring them to cryopreservation partner institutions in their countries, and they can be cryopreserved there.
Thank you very much. Adrian?
Yeah, and not only the new developments are necessary, all kind of samples, living samples, are necessary for biobanking and for preserving species. Then, if an improvement is reached for new technologies, it's better for some species, but we need all kind of samples if we are talking about conservation.
I would just share something about plants. Sorry if Adrian has done. Yeah. I would agree with all the comments made so far, also for the plant world, meaning that like in the tropics, we have like a lot of recalcitrant species worldwide, and many of them are particularly concentrated native to, the tropics, and botanic gardens in those countries might have, this challenge of dealing with a lot of recalcitrant or exceptional species, but might not have the resources for the, setting up cryo tissue culture or cryopreservation labs. And so definitely having, connections or collaborations with larger facilities, or those facilities that, where you can send the material and those species can be preserved. I think it would be a good idea. Yeah.
Thank you, Marcelo. Oliver?
Yes, building on what Dr. Mastromonaco said, there's opportunities for facilities that are practicing certain technologies to share information, to build alliances, to train others. And Dr. Heinemann, provided an example of that, and Dr. Mastromonaco did. So I think that the recommendation or the best practice will be to share expertise and procedures and training. And I'm kind of jumping onto what Adriana said, because that's exactly what the principle of this Latin American Biobanking Network is doing, is to have a mechanism to share expertise. And this then becomes benefit sharing that's called for in the Kunming-Montreal Global Biodiversity Framework. So it's a very tangible way for biobanks to inclusively and equitably contribute to building the expertise and the facilities and the capacity that's going to so benefit all of life.
Thank you, Oliver. I'm really being mindful of time, and so if nobody has any other pressing comments on that, I'd suggest that we stop that panel right now. And I would like to really thank you for all of you speakers, distinguished speakers, for taking the time today for 2 hours to be with us for this webinar, to share your experience, but also your expertise, most importantly, on those issues. I think it's very clear from what we've seen today that biobanking is a very important conservation tool that is needed and that will help us in achieving the targets of the global biodiversity framework, also from a fauna perspective, but from a flora perspective as well. And so we're very grateful for all of you to have spent time with us, sharing about your experiences and your expertise, and to all of you as well who have participated and showed great interest to this webinar. I will now hand over the floor to Megan Joyce from Reverse the Red for the final announcement. And from my side, I would like to thank all of you speakers and all of you participants for being with us today.
Echoing that, Lois, thank you all so much for this fantastic conversation. We had lots of additional questions in the chat and the Q&A, and so panelists, I might send you those so we can try to get a few additional answers out to our attendees after the webinar. We will send out our recording as well. And then I am also going to paste into the chat a link for additional upcoming Reverse the Red webinar sessions. We host them on lots of different topics. Today was focused on biobanking and cryopreservation, but upcoming we also have a tutorial around our Reverse the Red species pledge. So if you are working on biobanking or cryopreservation or any other actions related to species recovery, we would love to hear what that work is that you're doing. Learn more about the species pledge on July 7th. We also have a session around our upcoming World Species Congress and Reverse the Red Day celebrations. So if you're interested in getting involved there, hosting an event, a satellite event, an educational event, we'll have that information session later in July. And our next webinar is around funding long-term conservation impact. I saw a couple of questions around the resourcing needed for biobanking and cryopreservation, and a lot of our species don't recover in a year or two. And so what does that look like to really fund long-term conservation impact? So those are some of our upcoming sessions, and we are really glad that you were all able to join us for this time. And you'll also receive a link as soon as you close out this webinar to a survey. If you have other topics you'd like us to cover, please fill out that survey. Let us know what you thought, and let us know if there are any other topics you would be interested in hearing about in an upcoming Reverse the Red webinar. So thank you all so much, and we will hope that you have a great rest of your day and hope to see you all again very soon. Thank you.